is, how do differences in the microbiome kind of control drug response? And
Mm,
so, we got interested in this area, kind of similar to why we're excited about studying BHB as opposed to ketogenic diets. I think, when you think about pharmacology, it kind of narrows the scope to compounds that we know are really important for the body. And it kind of allows us you to then ask questions about how do differences in the microbiome affect either the level of those drugs or how they're metabolized and removed from the body,
mhm.
or
So
how they make it to the target
yeah,
tissue.
the general idea here is if you take a pharmaceutical drug, say the gut microbiome could be important for activating that pro-drug into the active drug. It could be important for how quickly the drug is metabolized and leaves your system, and all of that type of stuff.
Yeah, it's like, it's interesting to me, like the really back to the very early days of drug discovery, there were links between the microbiome and drugs. And so, for example, one of the first antibiotics that was ever developed is called Prontosil. It has what's called an azo bond, which is two nitrogens with a double bond in between of them. And those azo bonds are not readily broken down by our own body, but bacteria can act on them. And so, and then it turned out that the active compound in Prontosil is actually the metabolite. And so you
Hmm.
need a microbe to actually create the antibiotic,
Hmm.
even though the drug that's given is effectively inert.
Yeah, yeah.
Um,
There could be, you could imagine scenarios where, I don't know, maybe someone's doing a clinical trial, they're giving some drug to 100 people, and we'll just make up numbers here. Maybe it works for 50 of the people and doesn't work for 50. It could be that, what explains that is the gut microbiome and whether or not you've got the right bacteria to, say, turn the pro drug into the active drug.
right, yeah, exactly, and also in the opposite direction. So we have I mentioned digoxin earlier. That's a classic example in the textbook that is a drug that is not really thought to be metabolized very much by the host, but is metabolized by bacteria to a downstream compound called dihydrodigoxin, and that metabolite is no longer active. And so you can have microbes in the gut that are both turning on and off drugs in ways that could change what amount of drug actually makes it to whatever the target is in the body.
Hmm.
Um, and yeah, so that's something we're really actively working to try to unpack for various drugs of interest.
Well, we've already covered quite a bit.